Hey guys, welcome back. This is Lei. In this video, I'm going to show you how to confidently identify reproducible peaks in ChIP-seq data. A major problem of ChIP-seq data analysis is that peaks identified in a dataset are irreproducible. Unlike in R-seq data analysis, which has a fixed number of genes, in ChIP-seq data analysis, depending on person, tools, and the parameter setting, identified peaks can vary significantly. This tutorial demonstrates how to control this variation and generate a set of reproducible peaks. There are many ways to tackle this problem. The easiest way is to simply overlap the peaks across all the replicates. This is also the method we used for our differential binding analysis. Remember, we first created a consensus peak set and then compare their signals between conditions. However, in this case, our goal is to identify true peaks across replicates, so these methods are not appropriate. Another way is to poll before peak calling. Basically, we merge all the replicates into one dataset and call peaks on this polled dataset. This method has its pros and cons. For example, it increases the sequencing depth and simplifies the analysis workflow. However, there are more cons than pros. Not the best idea either. A better way is to use the IDR framework, the Irreproducible Discovery Rate framework. Here is the workflow. First, we call the peaks in individual replicates. The algorithm then rank and match the peaks between replicates. The paired peaks then go through the Copula mixture model. The IDR scores are then calculated. Thresholds are set based on the IDR scores. The pros of using this framework is its statistical rigor, and it uses signal strength. Of course, there are cons. This method requires consistent peak calling. That means the peak calling tool must be same, and it's less effective for broad peaks, and it only works on paired comparisons. That means it only works on 2 replicates. But what if you have more than 2 replicates? I'll show you later. Here is the illustration of IDR framework. Basically, we have 2 replicates, rep1 and rep2. We call the peaks in rep1 and rep2 separately. The IDR then takes the peaks from both replicates and generates a peak set with high confidence. If you don't feel confident enough, you can merge rep1 and rep2, split the poll sample into pseudo replicates, call the peaks, go through IDR, get a peak set, repeat this process for many times, and compare the number of peaks. If the numbers are consistent, you're good. If you're still not confident enough and have absolutely nothing else to do, you can split each replicate into 2 pseudo replicates, call the peaks, go through IDR, and get a peak set. Repeat this process for many times for each replicate, and then compare the number of peaks from the 2 processes. But seriously, what's wrong with you? Installing and using IDR are straightforward. On a Linux system, let's first activate our Homer environment we created in our previous tutorial. Then install IDR and its dependencies using conda install. Check its version. Call the peaks for each replicate using max2 call peak function. Why max2? Because IDR directly accepts the peak file from max2. Save your time for formatting. The example we use here is from our previous tutorial. Once we have the peaks from max2, here is how we use IDR. The inputs of IDR are the peak files from replicates 1 and replicates 2. The output is a peak set from IDR. You have to specify the name of the output. The input file type is narrow peak. All other parameters are optional. Here is the format of the output file. We have peak coordinates, and all other statistics like p-value, q-value, FDR values. For more details, you can consult my website. Here are the plots from IDR. Not much to see. Just remember, black is good, red is bad. A good result should have more black dots than red dots. What if we have more than 2 replicates? The key is to merge all the replicates into one single file and split this file into 2 pseudo-replicates and go through the same process. Here is an example. Assume we have 3 replicates, and we already have the BAM files. We merge all the BAM files using SAM2's merge. Then we split the merged BAM file into 2 replicates. Note, the output files are SAM files, so we have to convert the pseudo SAM files into BAM files using SAM2's, and then index the converted BAM files. And then we call the peaks for each pseudo replicate using max2call peaks. The called peaks then go to the IDR command to generate a peak set with high confidence. This is all we have for today's tutorial. Subscribe if you like it, and I will see you in the next video.